So, hello. Marco Mendoza couldn't make it today, so I'm stepping into his shoes as much as I can as his third year PhD student. And today I'm going to talk about one axis of my PhD research, meaning the characterization of the inflammation taking place in the colon of the colitis mouse models, especially using the on-house developed spatial transcriptomic assays. So to give you a little bit of more context, I'm a chief PhD student, meaning I'm working with both an academic laboratory, so the CISFAIT team directed by Marco Mendoza, and I'm also working with a French private firm, Novalix, where my supervisor is Anjigu, that is part of the pharmacological and drug discovery team, and they will provide us the tissue material in order to study the inflammation. So one axis of my PhD research is to study the inflammatory bowel disease. So that include the Crohn's disease and the ulcerative colitis. So both of them, they are chronic inflammation taking place into the colon tissue. And while the Crohn's disease is more an heterogeneity of the inflammation within the colon tissue, whereas the ulcerative colitis is more an homogeneous inflammation. But in both cases, we don't have currently curative treatments, mostly because we don't know the molecular aspect that is behind the heterogeneity that happen between the patients and within the patient itself. So in order to study EBD, the Novalix private firm, they furnish you some mouse models. So I'm working with the MDA1 alpha double knockout models. So basically, it's a mouse that will spontaneously develop colitis. And during the treatment, so 5 weeks, we're also going to expose the mouse to some bacteria in order to, at the end of the treatment, having homogeneous level of inflammation between the different kind of mouse we are working with. And in order to study the inflammation, we are going to use the in-house developed spatial transcriptomic assays, so the CISFAIT protocols. And we have published a paper in 2023 that give all the details about how to reproduce it in your lab and how is it working. And so at the end, when you produce the library and you send them to sequencing, you have to analyze it. And we also produce an on-house software, multi-layer, in order to do that. And you have a publication that explains how to install it and how to use it in 2010-21. So a short recap on how the on-house spatial transcriptomic assay working. The aim is to have on a slide a double barcode DNA that will be able to capture the mRNA within the tissue, but have spatial information. So we print it in 2 steps on microscopic slide. So the first step is to print one DNA array containing one barcode, so the raw barcode, and then a second step when we print the second drop of DNA containing the second barcode. And then at the end, with molecular reaction, we ended up with a unique double barcode DNA strand that will be able to capture the mRNA within the tissue. As you can see at the bottom, we have an image of the, at the end of the process of the printing process. So it's a control quality that each drop will capture, will have a unique barcode association. And as you can see, because it's a on-house and the printing process is happening in our lab, we can control the resolution and the size of it, depending on which kind of analysis you want to do and which kind of tissue you're going to work with. You've got a red picture there. So that's the picture that I've done with human brain organoids during the development of the protocols. So it's a biological scan, biological image of a tissue that is deposited on the DNA array area. And I don't know if you can see it, but we've got a red frame around it containing 3 layers of spatial or spots, red spots, that will help position the tissue and kind of guess which barcode we capture in the mRNA and how much area the tissue covers. And when we send them to sequencing, we are able to, using multi-layer software, we are able to reconstruct the numerical image of the tissue using the double barcode and the mRNA. And as you can see, you have the scan in red and you have the really, really similar shape of the tissue in blue. So I'm using these protocols applied to the study of EBD. So the perform they produce the colon tissue and then I record them. They are separated into 3 parts. So the proximal, medial, and the distal part of the colon tissue, they are fresh frozen in OCT in order to conserve the mRNA. And then I'm going to study them in 2 strategies. So the spatial transcriptomic one, and in order to have its own control and to control the quality of the data that we'll get through spatial transcriptomic, I'm doing also some pseudo-bulk analysis within the same tissue samples. Meaning when I'm going to cut with the crust as a tissue, as I'm going throughout the tissue, I'm going to collect section for spatial transcriptomic, but I'm also going to collect a number of sections in order to do mRNA extraction and then mRNA sequencing. And as you can see, I'm having different kind of sample of the pseudo-bulk mRNA as I'm going throughout the tissue and not just one samples. So the first step was to control the quality of my tissue. So I've got, I'll analyze the data and also I try to characterize them. So as it was just kind of a way to control the quality of my data, I mainly focus on the commonly expressed gene within my different kind of samples, of bug samples. And I'm using the mouse gene atlas public data using Enrich-R. And I'm quite happy because they characterize my commonly expressed genes as intestinal genes. Also, I wanted to use another public data, so the human protein atlas. And this time, I also use the commonly expressed genes between the 4 bulk samples. And I'm also happy to see that they are able to be characterized into the different kind of subcell type of the colonic cells. So that gives you it gave me a good quality of my samples. And now I wanted to link that to my spatial analysis. So you've got the recap of how I'm supposed to capture, how I'm capturing the mRNA within the tissue. So I've got mRNA linked to spatial coordinates. And as you can see in the red picture, this is a tissue that I've got. And I've got the frame in red that is helping me to position the tissue within the barcode and to see me, also to give me a first idea of the different kind of structure I've got within my tissue. And when I'm sending that to sequencing and then I'm visualizing the result on multi-layer, I'm able to reconstruct the quite same image on multi-layer. And I don't know if you can see it, but on the red picture, you've got some kind of vesicle architecture within the tissue, and I'm able to see that onto multi-layer by just doing a normalization. I haven't done any other analysis. So it is kind of a really good techniques. However, we encounter a limitation. It is a technical limitation linked to the printing device that we have, meaning the resolution. And because we still want to enhance the resolution of our data, we wanted to use a mathematical approach in order to counterbalance the technical limitation that we got. So we use a depixelating strategy, meaning from our spatial data that we got through multi-layer, we're going to take 4 neighboring pixels, making the mean of it, of the data, and creating a new pixel between them in order to enhance our resolution. And we control if it's working and if the quality of our data wasn't changing by that. And that's actually, it is good because you've got the, on the left side, you've got the original data. And on the right side, you've got the transformed data, the depixelating one. So we have a better resolution and we kept the structural information that we got at the beginning. But this is just like the number of the total gene captured in 1 pixels. And what we want to do at the end is to characterize the expression of a gene within the tissue. So let's take an example. So CNOT4, it is a gene that is involved into the transcriptional regulation factor. And it is, as we can see in the original data, already differentially expressed within the tissue. So in red, we've got area what is upregulated, and in green, area what is downregulated. And we were applying the deepscaling strategy. We have a better resolution, and the pattern of expression of this gene is not changed. It is enhanced. So it is we are happy with this strategy in order to enhance our resolution with the data without changing the quality of it. So to end my presentation, I would like to present to you some preliminary data. So using the same samples and using after the strategy of depixelating, the aim of all of this is to better characterize your tissue involving omics data with spatial information. So I'm using the matrix provided by Multilayer, where I have like gene expression linked to spatial information. And then we cross-link them with information from a paper, Garrido, that was published in 2023, when they characterize the sept cells, colonic cells that they found in a patient having EBD using single-cell technique. And where we crossed our gene linked to spatial to the gene character, the sept cell type characterized by gene, in order to have at the end, as you can see on the right part, the sept cell colonic type linked to spatial data. So that the aim of it is to better understand the tissue, better understand the how cells are within the architecture, but through the gene expression of it. And so I would like to thank you for your attention. If you have any question, I'm happy to answer it. And if we don't have time, obviously, I'm here until the end of it, so we can meet afterwards. And I would like to thank my team, obviously. So Marco, that is not here, but that helped me a lot, even with the biological aspect and the bioinformaticals, and all the team also. So thank you.
Thanks a lot for the presentation. Actually, you were already answering to my question at the very end when you say, if you understand correctly, you use signatures of different cell types in order to identify sort of the expression of the cell types for you and separate your spatial information. Have you thought of using the convolution methods like the ones that were presented before to try to understand where are the cell types per spot?
So I've actually seen the presentation this morning. So that's the point of me coming here, to have a better strategy and to have new perspective on how to analyze my data. Notably, I've done a tutorial with you on Tuesday. So now that I've got my data and they are good quality and I've got the basic strategy in order to analyze them, I would like to push that forwards and to use other techniques in order to really, really better characterize my techniques. So yeah.
No, okay. Hi. I have a related question. How many cells do you have in each spot, and how many genes can you detect with your spatial screening?
So we are talking here about like pitch resolution, right? So I've got here. So maybe I can move, but. So we've got here the center spots, right? And the spots is around like 100 micron wide. So we know like the cells are around like 10 micron. So theoretically, we are capturing like 10 cells, more or less. And by spot, we've got 200 million molecules. So it's a lot. So we can capture up to, I don't know if you see the data there. We've got up to 55,000 genes for 1 pixel attached after all processing. So it's quite a lot. I know that when we compare our own on-house developed protocols, even though the resolution is not as pushed at the one that you find in the commercial strategy, the yield of the final experience is much better. We've met on the paper published in 2023. We compare one commercial technique that was really present with our ours, and we see that the yield we have a good yield.
Thank you.